Спасибо большое. Good afternoon, everyone. Guys, sorry for some technical problems for the delay start, but we start now. And today is a very important topic about hepatitis viruses. Guys, not coming to the class, to the lecture. Guys, I don't see any students. So guys, microbiome lecture has started. Or Jasmine Sethi, good afternoon. Send everyone my message. We start already. So there were some technical problems. So good day Abhinav, Abhishek, and good day Vijeta and everyone. Start coming. Ishita, Rahul, start coming for a lecture. Okay, guys, we start now. But then at the very beginning, I would give some advertisement. So because of the complete lockdown of the university, we cannot take the checkpoints yet. So then we postpone checkpoints to the December because we have online studying until the 1st of December. So most probably we start with if we have very first offline week, then this week will be control week for checkpoint number 1 in microbiology and checkpoint number 2 number 1 in immunology. So that's why now we study plainly and we study viruses. So the next class is for viruses that you heard about already. These are the ortho and paramyxoviruses, and today's lecture is about hepatitis viruses. And this is a very large group of viruses that are really very important in human pathology. And when we had class for enteroviruses, we have discussed already or touched somehow already hepatitis A and hepatitis E. So today we won't talk about them. But then, taking into consideration that we have around 8 hepatitis viruses, we would like to present the common characteristics of hepatitis viruses, or rather diseases caused by hepatitis viruses. So irrespectively of what type of the hepatitis virus do we discuss, they can cause an inflammation of the liver. And quite often this inflammation of the liver is associated with the icteric symptoms or icteric stage of the disease. I hope you know already that icterus or icteric stage is the liver damage and release of liver enzymes and bilirubin into the bloodstream. So, and it usually accompanied with the discoloration of the skin, mucous membranes, the eyes, and so these are the signs of the jaundice. Then the second, all hepatitis viruses are readily spread because usually the infected individuals are contagious before or without showing the symptoms. What does it mean? That in the last stage of the incubation period, and I hope you remember that incubation period is a period where in between the entering of the microbe and the appearance of the symptoms. Within this period, usually patients are not infectious, but hepatitis viruses infections are the exceptions. So patients start to be contagious before the symptoms appear within the incubation period. So, and also what is important, some patients may have asymptomatic infections of hepatitis viruses, but even they won't have symptoms, they can be contagious. That's what we have here, right? And then all hepatitis viruses are different in their structures, in the mode of replication, and the mode of transmissions. And the specific cause, nature, and serology of the disease, cause and the disease they cause differ in each virus. So we will take a look at the viruses. Here you can see a table of the known by now or discovered by now hepatitis viruses. And here we have RNA viruses and DNA viruses. The only really very common DNA virus is hepatitis B virus found by Bloomberg in 1963-64 and then described by Dane in 1970. The others, we don't know yet whether they are exactly the hepatitis viruses or there are some modifications of hepatitis B virus. So we won't talk about them so much. Then we have more RNA viruses. And within here we have hepatitis A virus found or discovered in 1973 by Faiston. So hepatitis E virus discovered by Kuro 1980. Hepatitis C virus from 89, Hockton. Hepatitis D virus found by Risetto 1977 and hepatitis G virus found by Kim 1995. This is the last one really, except for this unknown very well now. So out of those hepatitis viruses we have studied already hepatitis A and E, but then I would like to draw your attention that the first hepatitis viruses found, as you see from here, were hepatitis A and hepatitis B. So these viruses first found A and B, and the rest of the hepatitis viruses for the very long while had been called as non-A, non-B hepatitis viruses, non-A, non-B. So quite often in clinic they still use this abbreviation to refer to the viruses that are not hepatitis A or B that can be easily diagnosed. So now today we take a look at the hepatitis viruses that are transmitted parenterally. So altogether hepatitis viruses may be classified into fecal-oral according to the fecal-oral transmission and parenteral transmission. We have studied A and E already. These are two hepatitis viruses that are spread through fecal-oral route. They are naked viruses both, so they are very resistant to the stomach acidity and the enzymatic activity in the small intestinal tract. So they stand this very well and will not be inactivated in the GIT system and stomach, right? While the parenteral hepatitis viruses, they will be inactivated in the intestinal tract. So that's why, what does it mean parenteral? It means these hepatitis viruses may spread by any route except for GIT. So, and we'll mention what are the basic routes of transmission, but then those are called parenteral transmission, is called parenteral transmission. So, so anyway, but not enteral transmission. Okay. Go ahead. And the most common hepatitis are hepatitis B, C, and D. And here we have the table where we compare the properties briefly of those hepatitis viruses. So the infection caused by B is called serum hepatitis. Hepatitis C is called post-transfusional hepatitis, and hepatitis D virus infection is called, or virus is called delta agent. So it is a defective virus. So it cannot cause infection itself solo. It needs some help. So we'll discuss it. And out of those, the only one which is HBV is double-stranded DNA virus, and C and D are single-stranded positive sense RNA viruses. So they have different taxonomical determination. So the hepatitis B fall into the family Hepadnaviridae and genus Orthohepadnavirus. Then hepatitis C enters the family Flaviviridae and the genus Hep C virus. And hepatitis D virus is not really very well classified because it is viroid-like virus. So out of them, all are enveloped by the surface glycoproteins are different. So for hepatitis B virus, the major surface glycoprotein is HBs antigen. In hepatitis C, we have envelope proteins 1 and 2. In hepatitis D virus uses the glycoproteins of hepatitis B to make the envelope. So it also is covered by hepatitis B surface antigen. They are of small to middle size. The smallest one is HDV because it's very small. So then hepatitis B virus 42 nanometers in diameter and C is 60 nanometers in diameter. So they are all acid sensitive. Then their prevalence in the human population is very high for B and C and is much lower and regional for hepatitis D. So now, what are the most important ways of transmission? For hepatitis B, parenteral and sexual. By the way, my dears, for hepatitis C, now we know that sexual transmission is very rare. It can be transmitted due to the sexual intercourse, but the incidence of these transmissions is very low. So the priority is given to parenteral transmission. So same to hepatitis D virus. Please pay attention that incubation periods of all those hepatitis viruses is very long. See, it takes up to 160 days or 180 days or 60 days for these viruses when they cause infection to develop symptoms. So that's why that is the duration of incubation period. So now it is understandable that at the end of the incubation period, these viruses, these patients infected, start to be contagious before they develop symptoms. So usually hepatitis B and C viruses cause the diseases with the insidious onset. And the only D causes the abrupt onset of the disease. It causes more like acute than chronic infections. So if we evaluate the severity of the diseases, so mild for both B and C, for B occasionally severe, and for C usually subclinical or very mild. But then at the end, after the several years of the subclinic or mild diseases, they may cause quite severe damage to the liver. So for hepatitis D, the infection is usually severe. So then, here the ability evaluated to cause the fulminant or very rapid lethal hepatitis. So the hepatitis B virus can cause fulminant hepatitis in 1% of the infected individuals. For HCV, it's extremely rare. And for hepatitis D, it's high to extremely high fulminant diseases. So when HDV combined with HBV, so usually the liver damage is very severe. Then now please compare, and that is a very important in the description of the diseases, especially in the difference between hepatitis B virus infection and HCV virus infection. So the share of chronic infections is different. So the hepatitis B virus causes chronic infections in 5 to 10% of the individuals who develop symptoms. We won't consider here the asymptomatic infection. So while the hepatitis C virus causes chronic disease in up to 80% of infected and symptomatic individuals. So that means that the acute disease is really, really rare for C, but quite common for hepatitis B. So then, in 10% of those who develop symptoms, the 10% causes cirrhosis in hepatitis B virus incidence, and up to 20%, even higher, for hepatitis C virus infection incidence. And please take a look also that hepatitis B is considered to be cancerogenic or oncogenic. It causes oncogenic transformation of the liver cells and primary hepatocellular carcinoma in 80% of those who cause chronic infection. And while the hepatitis C is also considered to be oncogenic for other reasons, and most probably it uses different mechanism of cancerogenesis, because hepatitis B is DNA virus and hepatitis C is RNA virus. But then still the cancer, primary hepatocellular carcinoma, can be seen in 5% of infected and symptomatic individuals. If we take a look at hepatitis D, it also causes cirrhosis, but most common is fulminant hepatitis. So vaccination, it is available against hepatitis B, but because hepatitis D virus cannot cause disease solo, it causes infection combined with hepatitis B only. So the vaccination against hepatitis B virus infection also protect us against hepatitis D infection. And unfortunately, there is no vaccine available against hepatitis C. Now, significance of the hepatitis virus. So by now we have more than 500 million people persistently infected with HBV or HCV, and are at risk of developing chronic hepatitis, cirrhosis, and liver cancer. And the inability to culture properly these hepatitis viruses. So it means those B and C considered to be not really cultural hepatitis viruses. So we don't have a good model to develop or to arrange the full life cycle of the virus, to learn it very well, and also to check the pathogenesis in the animal model. And the absence of cell culture system and the lack of very good animal model really limits our understanding of the virus life cycle and pathogenesis. And if we don't know this, so it's difficult to develop vaccine or chemotherapeutic antiviral drugs. So some breakthrough had been done in 2014, and that time a novel human hepatoma cell line, termed like you see here, has been established, that supports the entire life cycle of both hepatitis B and C. So it means we using these cells, we could infect them with hepatitis B or C, and these viruses will develop the whole replicative cycle with the release of newly synthesized infectious virus particles. So then, since this is done, that provides a powerful tool for addressing the virus life cycle and development of the antiviral drugs. Now, there is some breakthrough in the treatment of hepatitis C virus, and we will mention the several generations of preparation that are in common use right now. And but unfortunately, there are no good treatment developed for hepatitis B yet. So, but there are some drugs, candidate for the drugs that can be proposed for those infected with hepatitis B. So we go ahead with hepatitis B virus. This is our first topic today. So this virus infects liver, kidney, and pancreas of humans and chimpanzees. So and other animals cannot be really infected with this virus. So this is a quite small envelope DNA virus. And the genome is very small for DNA viruses, just 3,200 base pairs. And it has unusual DNA, unusual genome. It is circular, you can see it here, right? Double-stranded, but it is partly double-stranded. It means the positive strand DNA in this double-stranded DNA is incomplete. So that's why it is called partly double-stranded DNA molecule. And it is important that inside the virion we have an enzyme that is called polymerase, but this is unusual polymerase. So except for regular DNA dependent RNA polymerase activity, it can develop reverse transcriptase activity. What does it mean? It means this enzyme can act as RNA dependent DNA polymerase. So it can form the DNA chain using RNA as a template. This unusual activity, and the only one more virus has this activity. So the well-known HIV virus. So, and based on this sequence comparing, so hepatitis B virus is classified into 8 genotypes from A to H, and each genotype has a distinct geographic distribution. So, and you can see here on the electron microphotograph that hepatitis B virus forms so-called Dane's particle. This is a complete infectious virion, and the diameter is 42 nanometers. Why it is important? Because in the blood serum of the infected individuals, or in the blood, you can find also the subvirional particles. You can see here. See, here you can see the Dane particle, the infectious virion, completely done with all proteins and nucleic acid and all this. And you can see it here, right? So this is number one. Then you can see this spherical 22 nanometer in diameter subvirional particles. So these are number 3, right? So we don't have it here. This is something like this, 22 nanometers in diameter. Okay, like this, right? So, and they are empty. So there is no genome inside these subvirional particles. And also we can see the filamentous particles. So see here, number 2. And there are those also particles with 22 nanometer in diameter, but very long. They are also made up of the surface hepatitis B antigen and nothing else. So they are also empty. So these subvirional particles, number 2 and number 3, are non-infectious. It composed of just HBs antigen and lipids without viral nucleic acid. So they are non-infectious. But by the way, because they have the same viral surface antigen as the DANE particles, so they may act as decoy for antibodies. Antibodies may bind with those subvarianal particles instead of binding or apart from binding with the hepatitis B infectious virus particle. You can see this is the particles we are talking about. They are highly immunogenic, and they were taken initially around 30, 40 years ago as the antigens uh, processed into commercial vaccines. But my dears, now it is not done anymore. Since we found a lot of blood infections like HIV infection, we never use someone's serum as the vaccine. We cannot use it. So we go ahead. So we take a look at the DANE particle. It is a spherical and consists of lipid bilayer, and the hepatitis B surface antigen is inserted into the lipid bilayer. Then, if we go from outside to inside we have the icosahedral nucleocapsid in a nucleocapsid that consists mainly of the HBC antigen. This is a protein, hepatitis B core protein. It is called, C stands for core protein. So why do we call it antigen? Why do we call the surface also the antigen, my dears? Because this virus cannot be cultured. The antigens of the hepatitis B virus are widely used in the diagnosis. So, my dears, so there's no culture method used as a approach in diagnosis of this infection. The only antigens of the virus and antibodies against some viral proteins can be considered as important so-called serological markers of the disease, and the balance between these antigens or the presence these antigens and antibodies against them in the blood may help us not only perform differential diagnosis of hepatitis B, but also they may help to identify the stage of the disease and even the prognosis of the disease. So that's why all hepatitis B viral proteins are called antigens, because they are very important in the serological tests used to identify the used in diagnosis. Okay, except for hepatitis B core antigen, inside we have double-stranded, partially double-stranded DNA and the polymerase. So we call it pol-reverse transcriptase because it has reverse transcriptase activity. So, and this polymerase is covalently linked with the 5' end of the minus strand of DNA. So now we can see it in the cross section, right? So, and then except for hepatitis C antigen, we may have you have it here. We also may have the hepatitis B-E antigen. It shares most protein or amino acid sequences with hepatitis B core antigen, but hepatitis B-E antigen is processed differently. So, and it is not really, it doesn't stay in the infected cell. Usually hepatitis B-E antigen is released, secreted into the serum, and it will not be assembled in the virion. So, the difference is, if to simplify it, so that core antigen stays in the cell to make the new virions, and hepatitis B E antigen is released to outside. So of course, if we are talking about the serological markers of the disease, we cannot really find hepatitis B core antigen itself in the blood, but we can find hepatitis B E antigen in the blood. So it is used as important serological marker of the disease. The surface antigen that covers the subvirional particles and Dane particle, encoded by the same gene and read in the same frame. But we may divide it into small glycoprotein, medium glycoprotein, and L-glycoprotein. So the small contained in M, the M contained in L, and the large glycoprotein is an essential component for virion assembly. So these glycoproteins contain the group-specific alpha and type-specific DYWR determinants. What does it mean? I would like to remind you. What does it mean type specific and what does it mean group specific? The alpha glycoprotein or the alpha group specific antigen is same in all types of in all strains of hepatitis B virus. And those minor antigenic determinants are specific for the particular types of hepatitis B. So we said that it can be divided into particular types. So, and the combination of these antigens, alpha and one of the type-specific determinants, result in development of 8 subtypes of the hepatitis B virus. And we call them, for example, ADY, ADW, or somehow else. Now a few words about the replication of hepatitis B, my dears. The replication is unique. What are the unique features first? So, hepatitis B has a very, very special tropism for liver. And we will talk later on about the unique, not so far ago discovered receptor that is called sodium taurocholate cotransporting polypeptide, NTCP. So then, hepatitis B replicates through the RNA intermediate. Take into consideration that this is DNA virus, and it is unique way when DNA virus may replicate through RNA intermediate. And also the unique feature is that a lot of subvirional empty particles are produced, made up of HBs antigen, and these particles may be used or may act in a body as antigenic decoy for antibodies. So we miss one more protein of the hepatitis B that is called HBVX protein, HBX antigen. And it is believed that this protein is essential for virus infection and has been implicated in the development of the liver cancer associated with chronic disease. So that's why the finding of HBX antigen in the blood serum is a very poor prognostic sign. It means that most probably we will have the oncogenic transformation of liver cells. So this receptor that I mentioned, so here, this is the receptor. So the sodium taurocholate cotransporting polypeptide. It has been identified as a cellular receptor and also for satellite virus, HDV virus. Most likely, it acts as a major determinant for levotrapism at the entry level. So it means the virus uses this exactly receptor to enter the hepatocyte. You can see it here. So, what is the way of replication? To shorten it somehow, you may take a picture of this slide. So, because I just want to briefly discuss it. So, first it binds with the receptor, and you can see it here, right? Anti-CP receptor, and enters through receptor-mediated endocytosis. Then it performs the encapsulation, and the pregenomic RNA enters into the cytoplasm together with the polymerase, and will be taken directly into the nucleus. This is a very important property, right? So the DNA appear in the nucleus. And it is very important that first thing that the polymerase have to perform, so completing of the genome. So the first stage, that genome will be completed. So double-stranded DNA molecule, repaired, they call it, see, repairing. So, but then protein-free viral DNA undergo so-called covalently closed DNA formation. You can see here. And this is the no, sorry, I would like to describe it. See, okay. So this is covalently closed circular DNA, right? So this is a supercoiled, covalently closed circular DNA molecules. That happens due to the repairing of the DNA genome and aggregation of this genome. Why we are talking so much about these 3C DNA molecules? They are formed, that is the form in which this virus may persist in the host liver cells. And this DNA has a very long half-life. And by the way, they are not integrated into the cellular genome. But then we believe that they can be transmitted in this form, So from one infected cell to another infected cell. And the development of these 3C DNAs is connected with the using these cells as a reservoir for viral reactivation when antiviral therapy is interrupted. Until the viral therapy is performed, so it will compress or it will inhibit the replication of the virus. But then it is withdrawn, so the reactivation of the virus can happen through these 3C DNA molecules, and they act as a reservoir of the infection. Then what happens? So then these DNA molecules can be transcribed by cellular RNA polymerase into messenger RNA. See, the messenger RNA are performed here, mRNAs. And then from the nucleus, these messenger RNAs can be taken for translation into the cytoplasm on the ribosomes on the cytoplasm. So then, as a result of this translation, pregenomic RNA molecule will be enclosed into the newly synthesized core. So we have core assembly and RNA packaging. My dears, please understand that no DNA molecule they don't exist yet new DNA molecules. The RNA molecule will be assembled in the newly synthesized virions from those proteins together with the polymerase. Then what happens? So the reverse transcription activity of the polymerase catalyzes the synthesis of the negative strand genomic RNA here inside the core. See, the negative strand genomic DNA will be produced. And then from this moment, this capsid may again be decapsidated and the nucleic acid may enter the cycle. That is the propagating cycle. More and more DNA molecules will be produced. And on the other hand, this viral nucleocapsid can be taken and the maturation will go on. Do you understand this? So then what happens when the assembly and maturation of new virions are produced? So using RNase H activity of the same polymerase enzyme inside the nucleocapsid, a strand of RNA will be completely destroyed. And you see here, before the release, so the RNA will be destroyed and then positive sense copy of DNA start to be produced using the initial DNA minus sense as an example, as a template. And it happens together with the budding from the endoplasmic reticulum, where the surface glycoproteins are already inserted into the EPR membranes. So budding from those membranes, the virus then will be released, you can see here, by exocytosis. So then, once again, what are the very important sort of checkpoints? The repairing of the partially double-stranded DNA into complete DNA, double-stranded. Then formation of circular covalently closed DNA molecules, CCC DNA molecules, that may exist for a very long while in a cell. Then transcription from these 3C DNA molecules and translation the mRNAs into the proteins in the ribosomes of the cytoplasm. Then assembly of the nucleocapsids with the pre-genomic RNA and the polymerase inside, and then RNA pre-genomic RNA is used as a template to produce, for polymerase, to produce minus sense DNA. Then RNA will be destroyed and the positive sense DNA will be produced using minus sense DNA as a template, start to be produced. But usually there is not enough time to complete the positive sense DNA synthesis. So that's why by the moment when those DNA particles are released, their genome will be partially double-stranded. So the surface glycoproteins are taken from the EPR by budding, but then released from the cell occurs by the exocytosis. And also together with the DNA particles, a lot of subvirional particles also will be released. So now, my dears, on this scheme, we can see the targets to inhibit EBV entry and other stages of replication. So now several drugs are developed like Mircludex-B, cyclosporin A. They act as anti-CP inhibitors to prevent the binding of the virus. It doesn't work 100%. And also we can have the nucleoside or nucleotide analogs that can inhibit the activity of the reverse transcriptase. So, but this drug unfortunately don't give very good result in treatment, but this is at least something that can be used in therapy of the hepatitis B infection. So, integration of the hepatitis B viral DNA may be associated with a long-term infection and the development of hepatocellular carcinoma, my dears. So, what does it mean? It means that 3C DNA molecules can exist for a long while, but also integration of the genomic DNA, viral genomic DNA may occur with a human genome. So, and rearrangement and juxtaposition of the viral promoters next to cellular growth controlling genes occur, and that may cause the viral protein transactivation and the activation of transcription of not only viral genes but also cellular genes that may result in stimulation of the uncontrolled cell growth. That is the possible mechanism of the hepatitis B virus oncogenesis, integration with the host genome, and the activation or transactivation of the genome uh, expression, and as a result, stimulation of the cell uncontrolled growth. Now, pathogenesis of hepatitis B infection. You can see here multiple ways of how the virus may enter the body. First, we can see vertical transmission. So, it can be transmitted to neonates through blood. It can be also transmitted through intravenous drug abuse. And this is a vertical. So now horizontal transmission. So it can be through mother's milk, vaginal secretion, blood, semen, saliva, with any, so the virus will be shed with any of those liquids. So then, as soon as the virus enters the bloodstream by any of these routes, so then it will be circulating in the blood until it reaches the liver, and then the persistent infection will be established in liver. And here you can see the stages at which we may block the spreading of the virus and binding with a particular receptor by neutralizing antibodies. That's what we need as a result of vaccination. The neutralizing antibodies, if they are produced and circulating in the blood, they may spread that may inhibit or prevent the binding of the virus with the target liver cells. That is our task in vaccination. So then, when we have a liver infection, of course, we have viremia of the hepatitis B virus. It spreads again through the blood. So as a result of viremia, the virus may infect any body fluids, and any body fluids will be infectious. So then on that stage, if we have very good immunity, we may have the resolution of the disease. So, but then cell-mediated immunity will be critical in the killing of the virus-infected cells, but also antibody-mediated immunity. So, but then the problem is that the infected liver cells start producing a lot of hepatitis B surface antigens. And these hepatitis B surface antigens may bind with antibodies. But if antibodies are not enough to bind with them, so they form immune complexes and these large immune complexes may not be eliminated by macrophages properly. So then the presence of immune complexes, except for the liver damage, we may have so-called antibody-mediated diseases or immune complexes diseases as a result of hepatitis B infection. So we take a look at it. So the source of infection are any body fluids, route of transmission, injection into the bloodstream, sexual contact, or during the birth. So what is the character of the disease caused by hepatitis B? Usually it can be acute or in 10%, 5 to 10% chronic. So it can be symptomatic or asymptomatic, and the type of the infection or character of the disease will depend very much on the person's immune response. Usually incubation period is very long. You learn from the table from 45 up to 160 days, and also the duration of incubation period depends very much on the infectious dose, the route of infection, and the personal characteristic. So the infection proceeds for a long time without causing liver damage and symptoms because this virus is non-cytolytic and causes minimal CPE when while multiplying in the hepatocytes. But genome integration into the human genome may cause the latency and may cause the silent infection in the hepatocytes. So, cell-mediated immunity and inflammation caused by the viral infection. It may cause the symptoms, and cell-mediated immunity may be responsible for resolving of the infection. So usually resolution of infection allows the parenchyma to regenerate. It doesn't happen very quickly, but it takes half a year or even several years. But then in that case, the virus will be eliminated from the liver. But if we have insufficient T cell mediated immune response, then we have very mild symptoms, no icteric phase of this disease, and then development of the chronic hepatitis. So I think very well it will be demonstrated by this picture. So when the large inoculum happens, a lot of cells start to be infected and effective. A lot of antigens are released into the blood. So it's activate the immune system properly. So the very effective cell-mediated response will be produced. So we have a liver damage, acute disease with the symptoms like jaundice and release of the enzymes. But then as the outcome of the disease, most probably we have resolution of the disease. I mean, the worse, the better. If we have acute disease, then most probably we may have resolution because the severity of the disease depends on the effectiveness of the cell-mediated immunity. Is it effective? If it is effective, so then we have acute symptoms and resolution. Then another story is when we have very low dose of the virus. Then virus spread eventually from the cell to cell. Not so many antigens are released into the bloodstream if virus multiply very slow. So we have limited activation of cell-mediated immunity. So it result in the persistency of the hepatitis B virus particles in the liver. So most probably it causes very mild symptoms, but as a result, long-term chronic infection. And when we have the long-term chronic infection, most commonly the cirrhosis will be developed. So cirrhosis may also lead to the development of primary hepatocellular carcinoma. So, but then you can see here, if delta agent will accompany with hepatitis B infection. So, most probably we may expect fulminant hepatitis with liver necrosis and death of the patients. So, primary hepatocellular carcinoma is a well-recognized long-term sequel of hepatitis B infection. So, and it may happen endemically, and it is one of the three most common causes of cancer mortality in the world. So, and from 250,000 to 1 million deaths per year worldwide occurs as a result of primary hepatocellular carcinoma caused by hepatitis B virus. So it's a lot. So, two major causes of the chronic infection with the hepatitis B virus are the persistency of the hepatitis B virus as a result of this 3C's development, 3C's DNA development in the nucleus, and also the local immune tolerance in the hepatitis B infected liver. These are very important major causes of chronic infection. And here you can see the epidemiology of the hepatitis B, and you may take a look at, so the color of Industan Peninsula and the Russian Federation is same. What does it mean? So the prevalence from 2 to 5% in the population. So, but we have areas much more affected, which are the very red. So, with the prevalence more than 5% in the population. Okay, so who are at risk, my dears? People from endemic regions, babies delivered by chronically infected mothers, but also those drug users, so those who have multiple sex partners, those who require blood and pooled plasma products, those who are under hemodialysis, healthcare personnel that make contact with blood, and residents and personnel of institutions for the mentally retarded patients. So, if you take a look at the medical students, they are at risk. They are at risk because they usually very sexually active part of population. So they also have may have contact with the blood if they are involved in the medical instrumental some procedures, right? So, and of course, if they required some blood products due to some disease, so they also will be at risk. So then, laboratory diagnosis. We mentioned that the initial diagnosis can be done by clinical symptoms and the presence of liver enzymes in the blood. This is biochemical tests. And the biochemical test, this is, see, they are here, they're supposed to be here. Okay, see? So, the ALT/AST test, right? And the presence of bilirubin in the blood. But these tests will be seen in all acute hepatitis viruses infection, right? So that's why the differential diagnosis can be done based on this serology or serological examination or serological finding. So what are we looking for? First, we will look for HBs antigen and HBe antigen because these two antigens are secreted into the blood abundantly during the viral replication. So the HBS antigen is more general marker. So, but then the HBe antigen is very specific marker and is best correlate with the presence of infectious virus particles, and the person who has this in the blood is highly infectious usually. Then we may find antibodies. And the most important marker, the best way to diagnose a recent acute hepatitis B infection is to find IgM antibodies against HBC antigen, core antigen. Because the core antigen, I'd like to remind you, is not released into the bloodstream, but the antibodies against it will be really abundant in the blood, so we can find them. So, and here we can see the acute HBV infection. During the symptomatic phase of the infection, the window can be seen. So where the detection of AB surface antigens may be obscured due to immune complex formation. What does it mean? It means, my dears, when a lot of HBS antigen is produced, so it is produced and then it disappears. So, an antibodies against hepatitis B surface antigen may also appear very late. Why? So, this is the period when the HBS antigen and HBS antibodies will not be found. Why? Because they may form the so-called immune complexes. And by the way, the immune complexes are not detectable by regular enzyme-linked immunosorbent assay. So it means when we perform it, we don't see the HBS antigen or HBS antibody. So that's why it can be a problem. So except for HBS antigen, some other serological markers supposed to be used, the only HBS antigen and antibody are not enough. And the chronic infection will be seen in chronic infection. We will have the contingent finding of HBe, HBS antigens or both, and we may have the detectable or lack of the detectable antibodies to these antigens. That is the sign of the chronic hepatitis B infection. So now what do we know about the treatment? So take a look at it. So we have three possibilities. So if we have chronic hepatitis, so acute hepatitis is caused by hepatitis B virus is not need to be treated. So the only chronic hepatitis virus is need to be treated. So we have several possibilities. If we have hepatitis B virus, DNA and ALT is normal, and we consider the liver biopsy. If it has severe histological disease demonstrated, we need treatment. If we have no or very mild histologic disease, so there's no treatment. Then if the hepatitis D DNA is found by PCR and no active release over the ALT and ST, so we don't need treatment. So, but then when we need treatment is when the DNA of the hepatitis B is found, in ALT is very high, then we need to perform the treatment of hepatitis B, my dears. So, what is used for treatment? The prodrugs are interferons, but by the way, my dears, it doesn't give very good effect. Then nucleoside analogs are also recommended. And you remember, these are analogs that inhibit the reverse transcriptase activity of the hepatitis B polymerase. So direct antiviral effect. Usually it is combined with interferon or not combined with interferon. So, but still, the therapy over the chronic hepatitis B infection rather count on the uh, inhibition over the viral replication, but cannot really cause the release of hepatitis B or elimination of the hepatitis B virus from the body. So how about prevention? Here we have very good results. So you know that first the hepatitis B immunoglobulins may be administered as post-exposure therapy or for newborns delivered by HBs antigen positive mothers. And of course, screening of donated blood is performed to prevent the infection through blood products. Avoidance of the sex contact with the carrier of hepatitis B. And universal blood and body fluids precautions when dealing with the blood products. Sorry. And of course, vaccination is recommended. Now there's a routine vaccination for all infants and children and people at high risk groups that are not vaccinated yet. It is useful after exposure for newborns also from HBs antigen positive mothers and people who occasionally exposed to blood or secretions from the infected individuals. Immunization of mothers is recommended. Of course, I think all medical students are already vaccinated against hepatitis B. The type of vaccine is subunit vaccine. So, and the vaccine that we use right now produced by genetic engineering. So when plasmid containing the S antigen for HBs surface antigen is cloned into yeast Saccharomyces, and then when we culture Saccharomyces, they also produce the HBs antigen we collect, purified, and use for vaccination. So now we shift to hepatitis C virus because now because vaccination against hepatitis B is working very well. Maybe hepatitis C virus make more concern than the hepatitis D virus. So first, this is the virus from Flaviviridae family, and the genus is Hepasivirus, and where we have the only virus, hepatitis C virus, that infects humans only. So it is 60 nanometers in diameter, has an envelope, and the genome is the single-stranded RNA, and 6 genotypes are distinguished. And we may see the genomic RNA inside, right? And the specific capsid C protein, and then lipid bilayer with surface 2 types of glycoproteins E1 and N2 that make dimeric structure. Go ahead with this. Ideas. So up to 200 million individuals have hepatitis C virus infections. That represents around 3% of world population, including 3 to 5 million only in the United States. And here you can see the share of different genotypes. See, 6 genotypes are shown here. And mainly you can see that in North America, in Russia, then almost everywhere the genotype 1 is the most common. The second rate is genotype 3. See, here is very high. So, but this is rather endemic, right? And also 2 and 6 are very common. So, second and sixth. See, second and sixth. Also, second and sixth. So, altogether, first, then second, sixth, and third are most common. So genotype 6 is very typical for Africa. Oh, sorry, genotype 4 is very typical for Africa. It is not found anywhere except for Africa. So a bit in Scandinavian country and very small share in the North America. So according to WHO, regional office in the Middle East, Egypt has the highest prevalence in the world with 7% prevalence in adults of hepatitis C infection. So we go ahead. Okay, here you can see the structure of hepatitis C virus, and here we can see genome of hepatitis C virus, genomic RNA. And here you can see with different colors the proteins that are encoded by the genomic RNA. So, and the the translation is done of the gene altogether, and the proteolytic processing of the polyprotein is done by cellular peptidases, but by our own peptidases and viral proteases. And the viral proteases that perform the cleavage are NS2 and 3 and NS3. So, which breaks the polyprotein in the 10 functional proteins. My dears, this is a very important stage in the replicative cycle. Why it is so important? Because the process of proteolytic cleavage of polyprotein is a very good target for therapy to inhibit the activity over the viral proteases. That is very important. And finally, those cysteine proteases, serine proteases, and helicases are targeted with anti-hepatitis C virus treatment. Then we take a look at the P7 protein, have a function in, here it is, ion channel. It is important in maturation and assembly and the release of the viral particles from the infected cell into the environment. So, and non-structural protein from NS3 to NS5B from the viral form the viral replicase complex. So it means they are working as together as RNA-dependent RNA polymerase. And also this non-structural 5B protein forms the RNA-dependent RNA polymerase itself. So this helps in replicating complex formation. So now replicated cycle. Ideas. It also have some unique features to compare with other viruses. Hepatitis C virus exists as a lipoprotein hybrid lipoviral particle. See, lipoviral particle. That in which form it exists in the body. So because the virus particle is covered with the low density lipids of our own origin, of human origin. That's why, take a look at this particle. This lipoviral particle is not highly immunogenic. So that's why, so partially, that immune response is not really developed for very long while against hepatitis C, or it can be the number of antibodies, the level of antibodies can be really very low. So that is the problem with hepatitis E, very low immunogenicity due to the cover with the human low density lipids and very slow multiplication, and as a result very few antigens of the virus will be released and recognized by the immune system. And what kind of receptor is used? The so-called scavenger receptor class B, type 1, this is SRBI, you can see it here. This receptor, generally, what is the function of this receptor? It is very important in the lipid trafficking, and that's why the lower density lipids of this particle binds with this scavenging receptor. And then they also important in the lipid traffic. Then the one more receptor is very important. CD81, right? This here. It is used. So these two receptors can be used by the lipid particles that contains hepatitis C to bind to the human cell and to enter by receptor-mediated endocytosis into the clathrin-coated pits, and there's a very nice picture that demonstrates it. Okay? So go ahead. Then, so as an entry, we have RNA uncoating, and this is due to the fusion of the viral envelope with the endosomal membrane. And then we have uncoating, and nucleocapsid will still contain the RNA. Then RNA is released, hepatitis C positive sense RNA. It can be translated immediately into the polyprotein, and then the polyprotein can be or should be processed. So we mentioned already that it should be autoclaved by viral proteases and also human protease into the functional proteins. Then membranous web replication complex will occur, will be formed, where the viral assembly happens and maturation, and the core particles will be produced together with the newly synthesized genomic RNAs. Then they will be packaged, and then they will obtain the low density lipids as a cover and released into the environment by exocytosis. So, few words about the pathogenesis and spread of the hepatitis C. It's spread by the same routes: parenteral transmission, blood, needles, then sexual contact is really very rare, give really effective in transmission, and also perinatally. Viremia can be detected within first 3 weeks of the hepatitis C transmission, but then it may last up to 4 to 6 months in the people with an acute infection, and much longer, for example, for decades, when a persistent infection occurs. So acute infection is similar to acute hepatitis A or B infections. And in acute hepatitis C infection, the inflammatory response is not so intensive and the symptoms are really very mild. Fulminant hepatitis C diseases are very rare, but then finally virus can cause fatal disease. So chronic hepatitis is very often. So as we said, in 80% of those who develop symptoms, the chronic hepatitis occurs. So, and virus persists in the hepatocytes, and often this infection progresses to the cirrhosis. In 20% of chronic illnesses, the cirrhosis occurs. And liver failure occurs in 20% of those who have cirrhosis. And also, hepatitis C virus may cause primary hepatocellular carcinoma. How the lab diagnosis is performed. ELISA can be useful to identify chronic but not acute disease. Seroconversion occurs within very long period of time. So what is seroconversion? It means when the patient had no antibodies and then the patient is converted to seropositive in antibodies against hepatitis C. So PCR is the best indicator of the disease. So when we can detect the hepatitis C virus RNA in seronegative people. Seronegative, it means in those who don't have antibodies. And that is a very important. The most important, accurate, and sensitive method for diagnosis of hepatitis C infection is polymerase chain reaction. So, treatment and control. So, out of HCV infection, 30% is ecteric and 80% cause chronic hepatitis. So, if treated with interferon alpha, so the cirrhosis in 30 to 50 cirrhosis will occur, then it may prevent the development of the cirrhosis, or it may prevent the development of hepatocellular carcinoma from those who had cirrhosis. But now we have very good combination of the interferon alpha and antiviral drugs, because before we had only ribavirin and interferon alpha, but it doesn't help so much. Now we have first generation of the antivirals like boceprevir and telaprevir. These are both potent protease inhibitor. Do you remember? NS324A proteases inhibitor. So again, let's come back to this chart. What do we prevent? We prevent the cleavage of the polyprotein into functional proteins when we inhibit the protease activity of the viral proteases. See? This is a very good thing. So, and this combination with pegylated alpha interferon is very effective, to be shown, again genotype 1. And also we have already second generation of these drugs, sofosbuvir, and there's a nucleotide analog of the cellular uridine nucleotide that inhibits the polymerase activity of non-structural 5B protein. After ingestion, it is rapidly activated and taken up by hepatocytes, this drug, and incorporated into the RNA polymerase into elongated RNA primer strand. So it means the termination of the RNA synthesis will occur. This since 2013, this drug is approved for combination therapy against hepatitis C. And cimeprevir is the protease inhibitor, so it is also approved by FDR for treatment. So now, they claim that with the second generation of these drugs, the hepatitis C virus cure can be promised to 95 to 100% of patients. So what do we have with the control? So no vaccines are not available. Now, my dears, please remember, hepatitis B, No good treatment, but good vaccination, right? Developed. Hepatitis C. Now we have good treatment, but no vaccination. So, guys, unfortunately, just few words about hepatitis D virus. So we have 5 more minutes, please. Wait for 5 more minutes. So the virion is called delta agent because it is defective virion. It is RNA virus, unclassified into the family, but the genus is called delta virus. The virion is spherical, non-enveloped itself, but it should use the envelope of hepatitis B virus. So the virus is acid sensitive and prevalence is very low. Now you see the structure. So this is an envelope made up of hepatitis B surface antigen. Apart from this, it has hepatitis D or delta antigen as a core, right? And ribonucleic acid inside. So in fact, this is virus parasite of another virus, of hepatitis B virus. So usually infection is spread by the same routes as hepatitis B virus infection. So and usually damage to the liver result in direct cytotoxicity of the hepatitis B. My dears, hepatitis B and C are not cytolytic, while the hepatitis D is cytolytic by itself. And when we have the co-infection of the liver with hepatitis B and C, we have two ways of liver damage. Direct cytotoxicity by hepatitis D and immunopathological damage of the liver by the antibodies and TCTL against hepatitis B. So that's why it causes very severe disease. So the co-infection with hepatitis D virus increased dramatically the infection by hepatitis B. So, and fulminant hepatitis is the very often result of it. Usually, we have massive hepatic necrosis that can be fatal in 80% of cases of combined hepatitis B and D infection. The lab diagnosis of hepatitis D is done by ELISA or radioimmunoassay procedure. So there's no specific treatment for hepatitis D, and prevention is done by vaccination against hepatitis B. Because if hepatitis B virus doesn't multiply in the liver, there is no supply of hepatitis B surface antigen for HDV hepatitis virus. So that's why the one vaccine prevents from both types of diseases. So with this, my dears, I have to finish. So we don't know much about hepatitis G. So then very few. Okay, this is it. See you next time, my dears. Please, oh, the announcement. So please, the checkpoints will be postponed until December. My dears, please, next classes are going through the special virology. We go like this. Straight after enteroviruses, we study automyxovirus and paramyxoviruses in the same class. This will be the home task for the next week. Goodbye, my dears. We also have this announcement in VK. Goodbye.